Now we are coming to types of plant tissue culture. Okay, plant tissue culture generally how to make it we have studied so far. Now the essay I told you that is over. Now we are coming what are the different type of plant tissue culture? Organ culture. So we are taking one group of cells and we are developing some organs. Suppose we are taking a group of cells from the leaf will be developed or plant can be developed. So if organs are developed from plant tissue culture, it is called organ culture. Culture of embryos. Just before I told you embryoids. Anthers, only anthers will be developed. Anthers are the organs of a flower. Then ovaries, they are also present in the flower. Roots can be developed. And shoot means what? The main stem and the branches, that is shoot, or any other organs of the plant can be developed. That is called a organ culture. The second one is meristem culture. Now already I told you meristem. If you take the tip of the stem, a soft tissue is present, it is called a meristem. If you take the root, at the tip of the root, the same type of tissue will be there. That is also called a meristem. Meristem culture. Culture of meristematic tissues in the culture media, that is called a meristem culture. Now we are coming to another one technique. It is also based on the plant tissue culture technique. Now first we have studied about organ culture, second one is called a meristem culture, but the third one is protoplast fusion. It is another one major technique like tissue culture. Protoplast fusion technique is based on plant tissue culture. Now, how the protoplast fusion is taking place? We have to see. Normally, any plant, a higher plant, when it is reproducing, first the plant is producing flower. In the flower, anthers are producing pollen grains. Ovaries are producing ovules. Then fertilization will take place naturally. Pollen grains will go and fuse with the ovule. Zygote will be formed. Then the zygote will develop into seeds. Then when the seeds are well matured, when they are reaching the suitable place, each seed is developing into one plant, that is natural condition. But here it is not like that. Technically in the laboratory we are preparing new plants by one technique called what? Protoplast fusion. Fusion, fusion of two cells. Protoplast, just at the beginning of this topic I told you, protoplast means any plant cell you are taking, it is with a cell wall, cell membrane, cytoplasm and organelles, but remove the cell wall. If you remove the cell wall, the plant cell will have cell membrane, cytoplasm, nucleus and a cell organelles will be present. That is called a protoplast. Protoplasm, protoplast both are same. So protoplast means any plant cell without cell wall. Any plant cell without cell wall. That is called a protoplast. Suppose you are taking protoplast of one plant, one variety, protoplast of another one variety. We can make them to fuse together, so new varieties will be formed. That is called a protoplast fusion. This technique is meant for new varieties formation, hybrid varieties formation. Now we will take, this is one plant. The same thing, one more plant, entirely different plant we have to take. Now I will take one plant. This is one plant of one variety, one species. We are taking one leaf, that is X plant. And first we are sterilizing this X plant. First we have to dip it in water, we have to dip it in mercury chloride, so many things. Now we are dipping. That means what? All the microorganisms are removed. That is first sterilization of X plant. Then the leaf should be taken away, and from the leaf, from the X plant, we are isolating a group of cells, just like tissue culture technique. Inoculum. The group of cells we can call it as what? Inoculum. Now the inoculum should be transferred into the culture medium. Now this is the culture medium. But in the culture medium only enzyme mixture will be present. No other nutrients, only enzyme mixture. Because the group of cells what we have taken, each cell is having cell wall. We have not removed. Cell wall is present in each and every one. But for protoplast we have to remove the cell wall. That is the purpose, first one. So first the group of cells should be transferred into enzyme mixture. And we have to leave it for about some period of time. Now what is actually this enzyme mixture? We have to see. 0.5% myrozyme, one enzyme is there. Second one is 2% onozuka. Onozuka, it is another one enzyme. 13% sorbitol or another one enzyme called mannitol. Any one we can take, either sorbitol or mannitol, 13%. Now these are the three enzymes that are mixed. Why the three enzymes are there? They can dissolve only the cell wall. They won't dissolve the cell membrane, they won't dissolve the cytoplasm, nucleus or cytoplasmic organelles. So they are dissolving only the cell wall. So the three enzymes are called cell wall digesting enzyme. Cell wall digesting enzyme. The pH should be 5.4, the temperature should be 25 degrees C. In that pH and in that particular temperature, the three enzymes are removing the cell wall, digesting the cell wall. Now, in the beaker, group of protoplast will be there. The inoculum is having protoplast. Okay, the protoplast, what is given here, should be isolated, separated. Now, this is one protoplast, cell wall is removed. This is another one protoplast of another plant. Parallelly, we have to do that. Now, this is the protoplast of another plant. Cell wall is removed. Protoplast isolation by this method. Okay. Now, we are taking only the protoplast of two different varieties. Now, you are taking one beaker. You are taking one culture medium. I will tell what is this culture medium. Now, this is the protoplast of one variety. This is the protoplast of another one variety of plant. So, two different species protoplasts are taken. So, protoplast of two different species of plants. Okay, this cell belongs to one species, this cell belongs to another one species. Now the beaker is having one fluid, it is 20% sucrose. Sucrose is nothing but carbohydrate. So 20% sucrose is there. Now in the 20% sucrose, we have to add one more chemical that I will tell you. Now we have to make it so the cells will grow because food is given. Then in that one, that is the same one, we have to transfer. Here the sucrose solution is there. But along with that, we are adding one more chemical. Okay, that chemical is called a PEG. What is this? Expansion for PEG. Polyethylene glycol. The name of the chemical is polyethylene glycol. Plus calcium ions will be there. Here only sucrose solution is there. But here in the sucrose solution, we are adding PEG, polyethylene glycol plus calcium ions will be added here. So, the polyethylene glycol is fusogenic agent. It can make the protoplast to fuse. It will induce them to fuse. So, what will happen? The two cells, they are touching each other. First, the cell membrane and cell membrane will fuse. And slowly it will become a large cell. Then the nucleus present inside and nucleus, two nuclei, they will join together and form a large nucleus. So, single large nucleus will be there. This is called the protoplast fusion with the help of PEG plus calcium. Then the single cell, what is formed, should be transferred to culture medium. Now, we will take what is this culture medium. What are the things are present? First one is MS medium, Murashik and the scoop medium, taurursine diacetate. Then the light should be 1000 to 2000 lux intensity. Temperature should be 25 degrees C. If you maintain this in the culture medium, single cell, large cell is there, it is a fused cell, protoplast fused cell. That cell is keep on dividing by mitosis, multiplying by mitosis. One cell divides into 2, 2 into 4, 4 into 8, keep on doubling. After that, enormous number of cells will be formed. Yeah, big mass, we can see by naked eye. Now the big mass of cells, all the cells are alike, that is called a callus. Okay, it is called a callus. Okay, now you have to take it out, callus. Then the callus is undergoing de-differentiation. Now the callus is now considered as meristem, induced meristem. Now from the callus, new plantlets will be formed. New plantlet, either one plantlet, from one callus as such if you take, one plantlet will be there. Suppose you are dividing this callus into about 10 or 15 pieces. How many pieces are there? That many number of new plantlets will be formed. Now the new plantlet will not be similar to first type of plant, or it will not be similar to the second species of plant. It will be a new species. It will be the combination of the first species and second species. Since the combination is there, it is called a hybrid. Okay, it is called a hybrid. So new variety. This variety will not be seen like first variety or the second variety. It is a hybrid variety we are calling. So for the development of new varieties, the protoplast fusion technique, but basically it is a tissue culture technique. So that is a next application. Then the fourth one is cell suspension culture. Now what is the meaning of cell suspension culture? Now we are taking this. Now we are taking a beaker and in that culture medium is there. In the culture medium, a group of cells you are taking, but the group of cells are attached. Okay, now there are nearly about 15 to 20 cells are there, all are sticking with each other, but our purpose is here we have to separate them. Now that is called a suspension, we have to separate them. So what actually we have to do is, in the culture medium we have to take that mass of cells, then now after some time, after applying the technique, if you see, the cells are completely separated, the cell separation is taking place. How it is happening? In the equipment, one equipment is there in the laboratory which is called a aggressive shaker. Once the liquid is kept in that machine, now it is aggressively, it is a shaking, so making the cells to be separated. At the time of separation, one or two cells may be damaged, but we are not bothering about that. Maybe majority of the cells, they are separated fully. So one cell can be taken separately and they can do it for any other research purpose. So the mass of cells, we don't want mass of cells, we want separate cells, single cells. Now for that, cell suspension culture technique is used.